Welcome to Lecture 1 of Unit 7. We're going to start talking about hereditary genetics. So before we get started, I want to make sure that you understand that there will be mitosis questions on this test too. You need to be able to use the mitosis information in order to answer the meiosis questions. So let's get started. So the chromosomal structure of DNA is going to allow for the transmission of hereditary information following replication. And as we know, humans contain 46 chromosomes in each cell, or what are called 23 pairs. And each of these pairs is a set of homologous chromosomes. So basically what that means, homologous, is that they have the same gene. So one of the genes is from mom and one is from dad. And the position of each of those genes on that chromosome is referred to as a locus. So we're going to use letters in order to abbreviate the alleles found at each locus. So let's take a look here. We have got these chromosomes. Okay, here's our chromatids, replication occurs, and we now have homologous chromosomes. So basically what we're saying is that at one point on each of these chromosomes, let's say right here, this codes for eye color. Okay, so each one of these is going to have that gene in the same spot for eye color. So we know that mitosis is for replication of regular cells. Meiosis is going to allow for the passing of those 23 chromosomes out of 46 to the offspring. So let's compare a little bit about mitosis and meiosis. So let's review mitosis really quick. So we know it's going to provide a way to pass on the complete genome to cells, and it's going to be in the 2N form. So in other words, we're going from 2N and we create two 2N cells. We know that mitosis produces somatic cells, and they are diploid. And basically that means they're 2N. And so somatic cells are basically all cells except for the sperm and the ovum. The mitosis of one human cell produces two daughter cells, each with a full set of chromosomes. So again, going from 2N to two 2N cells. The process only involves one division, and it does not result in genetic variation. So the daughter cells are going to be identical to the parent cell. There will be no crossing over, no independent assortment, no variation at all. And this is basically what we went through before. So you've got your G1, your S, your G2, and then mitosis resulting in those two daughter cells. So let's talk about meiosis. So meiosis is going to ensure that each gamete is going to receive a haploid set of chromosomes, so just an N. And it is going to produce the reproductive cells, I can't spell, that are haploid gametes, so our sex cells, so sperm, egg, ovum. Make sure you're familiar with those terms, obviously. Meiosis of one human diploid 2N cell is going to produce 4 daughter cells. And each of those 4 daughter cells will have 23 chromosomes out of the original 46. So meiosis, this process is going to involve 2 divisions. They're very easily named, meiosis 1 and meiosis 2. And they're easy to remember because meiosis II looks very similar to mitosis. So meiosis is going to lead to genetic variation due to crossing over that occurs, the independent assortment. It also leads to random fertilization of sex cells, which increases the variation in the population. So if we look here, everything looks very similar to mitosis, except for this end result, there will be the 4 cells. So no matter what diagram you look at, if it only has these 2, you automatically know it's mitosis. If the end result is 4 cells, then you should know it's meiosis. Let's look at a little bit bigger diagram here. So let's talk about some of the variations here. So the first thing that I want you to look at is this is mitosis, right? And we know everything that happens in prophase, metaphase, anaphase. So the big thing here, difference-wise, is that metaphase, they line up in the middle. They kind of create this Congo line. Okay, so remember that. Something else to notice, the difference between the two, is that there's never going to be any X's here. Okay, so let's look at meiosis. So we've got basically 2 things going on here. So this first part here, we go from interphase 1 all the way down to telophase 1. Okay. Then we go into, and that's referred to as meiosis 1. Meiosis 2, everything is the same except it's prophase 2, metaphase 2. So the big difference to note up here, meiosis 1, it does look very similar to mitosis, but there are differences. For instance, metaphase right here. These are not lining up in the middle, and they're not being pulled apart the same way. So these are more like square dancing, whereas mitosis, metaphase, is like a Congo line. And again, same thing here in metaphase 2. This, we've got more of a Congo line versus the square dancing going on. So, again, the big thing to note is end of meiosis 1, we end up with those 2 cells. They go through replication again, division. We end up with those 4 daughter cells. Okay, so some points of emphasis that we need to make sure that you know. So, prophase 1 and crossing over. During crossing over in prophase 1, this happens between homologous chromosomes, not sister chromatids. That's important to note. So here we have our homologous pair. So we've got mom over here, we've got dad right here, or vice versa, however you want to label it. But one is mom, one is dad. And you can see that these homologous pairs, that the crossing over is happening there, not between sister chromatids. So that recombination is going to end up with variation. So this pairing up during prophase 1 is called synapsis. And the site where that information is exchanged is called the chiasma. So when those homologous chromosomes have synapsed and they form that chiasma, they are now going to be called a tetrad. So it's important for you to understand that crossing over is going to happen at different points. And sometimes it doesn't happen at all. It's very random. It is a random event. But it's going to lead to that variation in gametes that are produced. So during metaphase 1, the homologous chromosomes, they're going to align side by side. And during anaphase 1, the homologous chromosomes are going to be separated. It's important to note that in mitosis, sister chromatids are separated. So again, it's homologous versus sister. So there are 2 divisions in meiosis. Meiosis 1 is the first division. The homologous pairs are going to separate. And since the 2 cells arise and the chromosome number has been cut in half, this is often referred to as a reduction division. Then we have meiosis 2. This is the second division and looks very similar to mitosis. During metaphase 2, the chromosomes, they're in a single file line. And during anaphase, the chromatids are separated and you no longer have any X's. It's important that you know that for both mitosis and meiosis, the daughter cells will never contain any chromosomes that look like an X. So here again, this here refers to meiosis 2, this image, and you can see we end up with those 4 daughter cells. That is your key image-wise to always figure out which one we're looking at. Okay, so spermatogenesis and oogenesis. Spermatogenesis is the development of the haploid cell into a sperm cell. Oogenesis is the development of a haploid cell into an egg. And yes, that is the way that it is pronounced. It is oogenesis. Some people call it oogenesis. But all 4 haploid cells resulting from the meiosis become sperm, but only one of the 4 turn into an egg. So let's look as to why that is. So there is something called a polar body that they basically disintegrate, okay? So here, right here, let's take a look at spermatogenesis. So here you've got your 2N, the mitosis happens, meiosis 1, right? We go through meiosis 2, we end up with 4 cells. And we end up with 4 sperm cells. Versus here, in the oogenesis, we start out, we go through meiosis, right? There is a little bit of genetic information that gets pushed off to the side called the polar body. And the rest of the information goes over here to go through meiosis 2. And again, the same thing happens in meiosis 2. We've got a little bit of this information that comes off here to the side into a polar body. And so we end up with 1 mature egg cell. So basically, the reason why is if you look at the egg cell versus the sperm cell, if you compare the two size-wise, this sperm is very, very small compared to the egg. And the reason why is because basically that sperm cell, all it has is it's got some mitochondria in the tail, and this head right here only fits the 23 chromosomes that it needs to pass on. Okay? So it's very, very simple. Versus this egg, the egg has to have enough cytoplasm in order to have all of the organelles and different resources for the egg. So all of the organelles that come from or that are created when an egg is fertilized actually come from the mom. It comes from the egg. So that is why we only end up with this 1 mature egg, because it takes so much to make sure that there's enough cytoplasm and everything else in there to support the fertilization. Okay. Okay, so let's look at pathogenesis. And this is referred to as a virgin birth. And basically, this is reproduction from an ovum without fertilization. So the female self-fertilizes. And this occurs basically when a haploid polar body fuses with the haploid egg, resulting in a diploid zygote. So you remember we talked about just a moment ago all those polar bodies that come off of here. And they're supposed to disintegrate. But if they don't, and then they come here and they fuse with that fertilized egg, then, because remember, it has genetic information, right? So that fuses together and fertilization occurs. So it can occur as a normal process in some invertebrates and lower plants, but very, very rare in humans and other things like us. So it's important to note, though, that this that is produced is a clone. So there's no variation or anything like that. Okay, so mitochondrial disease. Let's talk about this. All cytoplasmic contents and organelles, including the mitochondria, come from the mom. So we talked about there's a bunch of mitochondria in the sperm, the tail of the sperm. But basically, it is just used to get that sperm on its journey, take it where it needs to go. Remember, we also talked about mitochondria contains its own DNA. So it contains the genes that code for those mitochondrial proteins. If we have non-functional mitochondrial proteins, it can cause mitochondrial diseases. So these diseases come from mom, and the severity of the disease differs from child to child because the ovum will only receive a partial number of the mitochondria. So what that means is that if you basically look here, and let's say that we have this oogonium that is going to go through mitosis, and in that we've got these mitochondria. Okay? And so we're going to say that this has the mitochondrial disease. When we go through meiosis 1, we're going to lose a polar body, which means that we're going to lose a mitochondria. So right here, we end up with the remaining 3. And again, we go through meiosis 2, we're going to lose a mitochondria. So we end up here with 2. Okay? So that is why it says that the ovum only receives a partial number of the original mitochondria. And this here is showing you the same scenario and what can happen. Let's move on and talk about variation. So variation in a population is very valuable in a changing environment. So some of the sources of variation in the sexually reproducing population are point mutations. And this is going to be due to a replication error. It could also be due to chemicals, viruses, radiation, heat, lots of different things. So, mutations that arise in germ cells are going to be inheritable, but most mutations do not lead to good variations, but there are some that could result in some advantageous traits. An example of a disease caused by a mutation, a point mutation, is sickle cell anemia. So over here, this image, we have sickle cell anemia. So basically, all that's happening is a single base gets changed during replication. And so you can see right here where we start out with an A, which pairs up with a T. Over here, somehow during replication, that T, or that A, was replaced with a T, so it reverses down here to where we have an A instead of a T. So our RNA code is different, which means that our protein is different. The second source of variation is from crossing over. And this happens during prophase 1 of meiosis. And basically, we've looked at that a little bit, but the homologous chromosomes, they're going to exchange parts, right? And it happens differently each time or not at all. So as I said before, it's a very random event. So there's no predicting where it's going to happen or how it could happen. Our third source of variation is the segregation of chromosomes and the random independent alignment of those chromosomes during metaphase 1 of meiosis. So the segregation of the homologous homologous chromosomes means that you will only pass on one of your two chromosomes. So more or less, if you look, the big G, it's going to be segregated from the little g. So if you look here, we start out with all of these, okay? We're going to divide that up. You've got two big G's here, two little g's there, and you will end up with only one big G or one little g, and you will only pass on one. So the independent assortment means that the chromosome pairs are going to line up independently of each other. And this is referred to as the law of independent assortment. So with 23 pairs of chromosomes all lining up differently during that meiosis, one human has the ability to produce 2 to the 23rd different, I can't do that exponent, so I'm just going to change colors here so you know, 2 to the 23rd different gametes. So that means 8.4 million different gametes from a single person. That's how many possibilities there are. Our fourth source of this variation is going to be random fertilization. So it's going to add to our variation because any sperm has the ability to fuse with any ovum. So the fusion of the gametes, each with their own 8.4 million possible chromosome combinations due to independent assortment, gives humans 70 trillion diploid combinations that can arise due to fertilization from two people. And that's just from independent assortment. So that 70 trillion does not even factor in the crossing over that can occur and producing different recombinant chromosomes. So that number, I mean, it's astronomical and it's amazing because no two zygotes will ever be the same. Our fifth source of variation, this is going to be from chromosomal mutations. And these are going to be different from point mutations. So make sure that you understand that. Point mutations is one base. So chromosomal mutations is basically when chromosomes break during meiosis and then they're rejoined, and sometimes it happens incorrectly. It doesn't usually lead to a good variation, and it can actually result in physiological as well as developmental abnormalities or syndromes. So there are several types. We have A, which is deletion. So basically, the chromosome is going to break, and that segment is completely removed. It's not added back in, so you can see we are supposed to have A through I. Now we go A through A, B, C, and then GHI. B is inversion. So this happens, basically the chromosome, it breaks, and the segment is put back in, but it's put in upside down or it's inverted. Then we have C, we have translocation. So again, the chromosome is going to break, and the segment is attached, it's reattached, but it's reattached to a non-homologous chromosome. So that's what's happening here. Then we have duplication, and this is when a segment of the chromosome is in more than one place in the chromosome, and it's due to replication mistakes. So you can see we have A through I, but then right here we've got F, G, H, I added back on. So an example of this duplication is Huntington's disease. Another example is of a chromosomal variation is Cry-Duchat syndrome. And basically you can see that part of 5, it's a little bit hard to see, but part of 5 is missing. And it is actually added down here to 8. And so this is an example of translocation. Our sixth source of variation is going to be caused by non-disjunction. And it's going to lead to what is called aneuploidy. So aneuploidy is a change in chromosome number, and it's different from the normal by 1. So we know that we have 46 chromosomes as humans. So that means that if you have aneuploidy, you either have 45 or you have 47. So there's a couple different types. There's monosomy, which is when an organism has only one of a particular chromosome instead of the normal 2. And an example of that would be Turner syndrome, which is shown right up here. Number 14, you can see is missing a chromosome. And then there's a trisomy, and this is when an organism has 3 of a particular chromosome instead of 2. And that can be seen right here. This is Down syndrome, and it's also referred to as trisomy 21. In animals, this non-disjunction does not usually lead to a good variation. It can result, again, in the physiological and developmental abnormalities. And here are the examples that we just talked about, the Down syndrome, the Turner syndrome, and then there's also Klinefelter's. And what happens in Klinefelter's is that a male actually gets an extra X chromosome. So they have 47 chromosomes. So all aneuploids are caused by non-disjunction, which occurs during meiosis. And it occurs when the homologous chromosomes fail to separate during anaphase 1, or it can also occur when the sister chromatids fail to separate during anaphase 2. So this shows you here non-disjunction during meiosis, and here is normal separation. Normal separation has occurred. You can see over here, this is non-disjunction. We've got nothing there. So that separation did not occur. And you can see that it can happen in multiple places. Here, this is where it's happening in meiosis 1. This example is where it has happened in meiosis 2, or as a result of the non-disjunction in meiosis 1. And here's just another image showing you the same thing. So polyploidy is when the genome has been doubled due to failure to divide. So in plants, polyploidy is very common. It can lead to new species, actually. And it's going to increase variation in biodiversity. Scientists actually think that 30 to 70% of today's angiosperms originated due to polyploidy. So basically what's happening here is that you've got the parent, you've got replication occurring, okay? And then you've got these tetraploid cells, which this image isn't very good because, of course, these should be joined right here in the middle. So I'm just going to draw those in. You can draw them in if it bothers you also. So replication occurs. We've got our tetraploid cells. And then there is failure of division. Failure occurs, and then they end up self-fertilizing. So they end up with those 12 chromosomes. They end up as 4N. So, in summary, how does normal variation arise through sexual reproduction? So, we've got crossing over. We've got independent assortment. And we've got random fertilization of gametes. And how do abnormal variances occur? We have point mutations. We have chromosomal mutation. And we have non-disjunction or aneuploidy that can occur. And that brings us, my friends, to the end of Lecture 1. And I will see you next time.